Thank you so much. Can you see my slide?
Yes, we can see your slide very well. Thank you.
Okay. Thank you so much for this for Julie, of course, the team to organize this very exciting workshop. I would like to acknowledge funding from NIH, 2 different programs, also funding from Separate in Texas. I just want to first give a very brief introduction of the gene editing. The idea is that you use an engineered nuclease to generate a DNA double-strand break at a user-defined site. That's where you define. So once that happens, there are 2 major DNA repair pathways. The first one is non-homologous end joining, which usually induces small deletions, inserted mutations, or indels. But that could be very useful. For example, groups have been using this approach or pathway to do gene disruption, for example, BCL11A. So the second major pathway is programmed repair or homology direct repair, or HDR, that requires a donor template. And we have been using this pathway to correct the sickle mutation in HBB. There are 2 major methods to do therapeutic genome editing. The first one is ex vivo gene editing. The basic idea is to say if you have a sickle cell patient, we isolate CD34 positive HSPCs, then deliver a CRISPR Cas9 and the correct donor template into those cells. So after gene editing, we put them back, and those gene edited cells should produce healthy red blood cells instead of sickle cells. However, we do have some challenges. In addition to off-target cutting, some on-target deletion, which I will mention, optimize engraftments and also high cost. Those are still potential issues for translating this into clinical practice, especially in Africa, as both Emily and Julie mentioned. So alternatively, we could do in vivo genome editing, but there are a lot of challenges in terms of delivery, tissue specificity. Even if we do in local injection, there are still some issues. So just quick summary of what we have done over the last few years. We have systematically optimized CRISPR guide RNA design and also the design of the corrective donor. We edited CD34 positive HSPCs from patients. We can achieve pretty high efficiency of gene correction. We also perform the genome-wide unbiased off-target analysis. Using a high-fi Cas9, we can significantly reduce off-target effects, which I'll show you. We also demonstrate a good level of engraftment. So just show you some data. So those results here show the gene editing outcomes from CD34 positive HSPCs from 5 patients with sickle cell disease, and each point actually represents the result from a individual. So you can see that the gene correction rate showing here is pretty high. From a particular patient, it's about 37%. Of course, with others, a little bit lower. So this group is really to show the cutting occurred, but there's no gene correction, meaning there's no donor integration. As always, we still have a fraction of the cells that have no cutting. So here shows if we differentiate gene-edited cells to red blood cells, of course, with control, as always, like around 20% of the cells are sickle cells. This is no gene editing. So with gene editing, this percentage of sickle cells reduced to around 1.3%. So that's good news. So molecularly, we did analysis of the the expression of different hemoglobin molecules. Again, in control, you can see the red shows HPS. So that's a signature of sickle cell disease. This patient was under transfusion therapy, so there's a little bit HbA, but otherwise you can see dominantly that's HBS. So with gene editing with a donor template, you can see the red part, which HBS reduced dramatically. The HbA, that's the normal hemoglobin, increased dramatically. The blue is actually the level of fetal hemoglobin. There's some increase. What surprised us was this, that the cells that in this case only had cutting, meaning there's no donor integration, but there's a pretty large increase of fetal hemoglobin. So as I mentioned, if we do gene editing, the three different subpopulations, gene corrected, cutting, no gene correction, no donor integration, and the cells without cutting. So this portion shows with cutting but no donor integration. But if this is indeed true, we are still doing more experiments also to understand why this is the case, but this could be also very beneficial. So these two combined, this subpopulation and the gene corrected one combined would be something like 80%, so we do not need to do selection to have a positive outcome. So Emily mentioned the issue of off-target, which is very important because we know that CRISPR-Cas9 cutting induces off-target like small deletions, insertions, or indels, but also could have large deletions and insertions. So recently we found at the on-target site, there are a lot of large deletions. It's around 10%, so it's not like a very small amount. So this is ongoing work. So also, if you have both on-target cutting and off-target cutting, that could induce large-scale chromosomal rearrangements, such as inversions showing here, large deletions, and also translocations. So again, that's an area we are still working on. So because we are trying to modify sickle cells, if we have even very rare events of chromosomal rearrangements in a small number of cells, that could actually be a big issue to induce disease. So just to show you for treating, again, the patient HSPCs, so the blue bars here showing the true off-target effects, because in this case, we first used a bioinformatics tool. We identified 57 potential off-target sites. Then we used the deep sequencing to quantify the off-target cutting, and we found out those are the ones that had above 0.1% of the cutting rate. Now, with a hi-fi Cas9 that was developed by IDT, and both Matt Porteous and I, we were, on my lab, were part of the team developing this hi-fi Cas9. So those are showing as the red bars. So in particular, this log scale. So this particular site called off-target 18 induced a high level of off-target effects. And here shows that with hi-fi Cas9 compared with wild-type Cas9, there's no change of on-target editing rate. Now, here again shows the blue bars are with wild-type Cas9, the red bars are with hi-fi Cas9. You can see again this log scale. the inversion and the deletion, those are chromosomal large-scale inversion deletion, all reduced dramatically in this log scale. So now, as Sienna mentioned, it's likely that off-target effects depend on individual's genome. We are developing a new bioinformatics search tool to identify off-target sites, but considering the individual's genome. So for engraftment, we did 2 rounds of engraftment. So the first study, we edited peripheral blood CD34 positive cells from 5 patients. So we injected into the immunodeficient mice, and we got roughly 30% of engraftment rate. In the second study, we took gene-edited bone marrow CD34s. So we got roughly 7.5%, which is lower, which is actually typical, because in this case, we did IV injection instead of direct injection. So we actually working with Julie's lab group and also a group in France. We plan to do more extensive engraftment studies with patients from different places, with cells from peripheral blood, bone marrow, and cord blood. So as you can see from this slide, if we perform gene editing by delivering CRISPR-Cas9 with donor template into patient HSPCs, there are 6 different outcomes possible. And for example, we could have both alleles gene corrected, or none even cutting, and or one allele gene corrected, the other allele just original mutation. So in any case, with the cutting, there is a possibility of inducing beta thalassemia, and we are still looking into that. So also, as I mentioned, on-target cutting could induce large deletions and complex rearrangements, and which may have pathogenic consequences. That's something we are still working on. So just switch the gear because of the high cost, because of the complexity of the approach I just mentioned, that requires very likely pretty complex procedure. So ideally, if we just do injection, let's say just 2 injections, if we can cure sickle cell disease, that would be fantastic. So in that case, for in vivo gene editing, I think perform gene disruption instead of also delivering a donor template for gene correction. That, I guess, compared with that, I think gene disruption in vivo is a logical choice. But we need to achieve both high delivery efficiency and high editing efficiency. If we use a viral vector like AAV, that's what we have been doing, we can achieve pretty high delivery efficiency, but for now, there's no good way to control expression. The CRISPR-Cas9 can be expressed for a long time, 9 months, a year. So that could induce genotoxicity and also immune response, which I will show you. So the current non-viral delivery, including using nanoparticles, the efficiency is still relatively low. And also there's no good control of tissue specificity, but although we could targeting. So one strategy is to do targeting with AAV, and which for example, in this case, AAV packages CRISPR-Cas9 for gene disruption. For example, we have a ongoing NIH-funded project to develop a targeted AAV. So I guess if we mobilize CD34 positive HSPCs, then we can do systemic delivery, although there are challenges there. also the question, can we just do local injection into bone marrow? Again, this is just a question. So we have done recent work to examine pre-existing immunity to Cas9. So Matt Portier's lab and another lab in Germany, they have done outstanding work to show 78% of humans exhibit an immune response towards both SA-Cas9 and SP-Cas9. In addition to neutralizing antibodies, many individuals also have T-cell memory against the Cas9. So what we did was we delivered AAV8 vectors packaging SA-Cas9 with a guide targeting a particular gene. So we first actually pre-immunized mouse against SA-Cas9. Then we found out there's a pretty large increase of CD8 positive T cells, and over a 12-week period, all the gene-edited cells were removed, killed by T cells. So just quickly show some data. So this is really just to show over a 2-week period, CD8 positive T cells increased by a few fold. This is really to quantify that. That's an indication of immune response. Then, so the two very clear indication, one is the genome, because we also delivered a vector just including GFP. So here shows the viral genome actually by 12 weeks reduced by 97%.
I'm sorry, Dr. Mao. Can I invite you to wrap up, please?
Yeah, just one slide. Yeah, one slide. Yeah, this is pretty my last slide. So then also for the vector, including the gene editing, the viral genome, we tested also reduced like 96%. This is really to show the indel, the indels in the liver tissue, all these are in the liver, reduced dramatically. You can see by 12 weeks, pretty much all gone. So in summary, ex vivo gene editing for curing sickle cell disease has a great potential, but engraftment needs to be further optimized, and on target site, large deletion insertion need to be carefully studied. So again, as I mentioned, cost is the issue, and also requires good infrastructure and training, as Emily mentioned. In vivo gene editing for curing sickle cell disease in Africa has significant challenges. As I mentioned, high delivery efficiency, low cost, tissue cell specificity, and immune response. Of course, off-target gene editing and off-target tissue editing remain major issues. So with that, I would like to acknowledge my collaborators. I have been collaborating with Vivian Sheehan at Baylor. She will give her talk, and of course with Matt, we started collaborating in 2008. We still have a lot of collaborations. The immune response was in collaboration with Bill and Christine at Baylor. I should mention that we have been working with Julie's group since about a year now, since last year. Thank you so much.